I wanted to do a couple of examples sort of exploiting the detail search fields, and these are actually inspired by you. We asked you for input prior to the workshop for things that you might be interested in doing in terms of queries. So, there's one Tcell and one Bcell, and at least two of you will find this enjoyable, I guess. So the overall goal is that it demonstrates query strategies that obviously can be applied in larger contexts, but they are specific queries. The first one being a Tcell one, asking the question to find Rift Valley fever virus T-cell epitopes tested specifically on CD8 T cells and defined in humans. So we have a little bit of everything. We have a source of interest, we have effector cell phenotypes, and delimiting to a certain host. So again, as I had pointed out early on, their specialized searches are available from the home page, from the drop-down menu, and in this case, we're going to do a T-cell assay detail search. So by clicking that, remember you're brought to this page, and as was raised a moment ago, as Carrie pointed out, what you see up here is an unfiltered view of basically all of the detail. And what I think we haven't been good enough about pointing out yet is that the filters as they're added, or the search criteria as they're added, whether that be through this interface or through the homepage interface, are going to be listed one by one up here next to the filters kind of designation along this toolbar. Very similar to what you would see like on an e-commerce website, online shopping, where you say, I want red shoes, I want leather only, and then you can one by one add or subtract those. So for instance, in this case, I'm going to use the search fields on the left side to define the source of the source organism of the epitope. And again, in this particular scenario, what you see a screenshot of is just doing the Google-style autofill and using the suggested organism that comes up. But of course, the finder could be employed as well to see like a hierarchical view or see related species around that, choose higher nodes, as Carrie had pointed out earlier. But in any case, when the organism is selected, you see right away that
Can you go back just a second?
Sure, I'm sorry.
Admittedly, this was my example, so I have a vested interest. So if I wanted to select all three of those,
all three of these, okay.
Yeah,
so
and are they different from one another in the data they're going to output?
Okay, great. So first, there are two ways to select all three of those. This is a multi-select autofill. So when you type that in and click on the first choice, you can again put your cursor back into the free text field and search again. Otherwise, by opening the finder, you could select one by one within the tree. Now,
also, Nima, I think in this case, can you maybe show it? I think at the time, right?
Thing. Oops. There we go. Okay.
Can you go to the finder? Because
Sure.
actually, I think the top one is probably all three.
Oh, if there's the tree, you mean?
Yeah.
Okay, so here we have kind of the most immediate hits, and Carrie had pointed out earlier that this icon means highlight that specific row in the organism tree. So as I click that, you'll see the tree opens up. And there, as Bjorn mentioned, this is a parent node of these two which have specific strain information. So the quickest way from the tree to query for all three would be to simply select this, and you see it sort of the theme pervades with the populate or the choice that you set as a filter popping up here. Similarly, you could select individual ones, one or the other. And to address your question about what the difference would be, what that means is that the curation, the particular record, would have cited those strains, and the paper would have, and so would have we. So if you choose one of them, you will get the one that cited that particular strain. If you choose the high node, you will get all of them, in essence. So definitely a more broad query if you have no, if you're kind of agnostic about strains, you just want all the information, the high node is definitely the way to go. It's going to be all inclusive. Good question.
So, and if you had chosen that so if you go back to the autocomplete.
On the regular page?
Yeah. So the first one of those, the first one is the high node. So the moment you search the general one, it should normally be on top.
Right, good point. Yeah. So we have devised these to be sort of intelligent in the order that they appear, so that the higher node is the first suggested one. But you can always confirm that by the IDs as well within the tree to see that there's a one-to-one relationship between what you're seeing here and what you see in the tree. Any other questions about that? Anything so far? Okay, so I will switch back to the screenshot view. Okay, so and then simply kind of repeating the same idea where we drop down to the next set of fields where we can define host information, Homo sapiens here. Then this is a nice way to kind of illustrate the assay cluster of fields that I had pointed out earlier. In this case, under the effector cell plus sign, I've opened it up so that you can see some of the fields that are available. And under the actual effector cell type, this is a drop-down list where we have a whole host. Here you see a fraction of them, but you can scroll down and find the type of cell that you're interested in. These are multi-select fields, so you don't have to limit it to just one type of cell. But in this case, I've chosen only the CD8 positive T cells. Incidentally, some of you I think a couple of people have asked about TCR information, TCR phenotyping. Just below that is another set of fields called assay TCR molecule. These would be cases where the T cell receptor has been sequenced and the paper gives those. That's not information we encounter frequently in the publication. But when we do encounter it, if there's an epitope-specific T cell and the TCR sequences are known and published and we can tie them into GenBank, those would be found there. In any case, you can see there are many more fields. For example, culture conditions. If you were interested in cells that were not treated in any way prior to assay, for example, not re-stimulated, not clonal, but directly assayed ex vivo, you could specify that. And again, we can show you more detail on these in the live queries. But moving along, sorry, my mistake. Moving along with the this particular query, if we hit search, we then see the standard kind of results separated by the tabs. In this case, we have 61 epitopes. But you can see pretty consistently found across the nucleoprotein. I'll get this right eventually, I'm sorry. Okay, so one of the kind of beauties of the database is that, like I mentioned in a sort of e-commerce situation, you can mold your queries or refine them on the fly as you see the results come to you. So if a lot of times you might have a somewhat open-ended query and you see the data that you get from that query, and then that might shape another question that you want to ask. So in this case, if we've satisfied the kind of first check mark of finding those T cell epitopes, but you now want to take that query one step further and say, now have tetramers been made with any of these epitopes, shown to work? You could simply just kind of pick up where you left off with that previous query, but add more stringency to the query parameters. So that's what I've done here. This is that results page that we were just on. And as you've seen now, the assay finder and or the assay autocomplete in the bottom assay cluster of fields can be used to specify specific assay types. And in this case, by typing in the word tetramer, you can get the result of interest or the choices you're interested in. In this case, I chose the tetramer assay. And remember that anytime you modify the query parameters here, by choosing the modification or the extra filter, you don't automatically filter the data. You have to re-execute the search. So what you'll see is when you modify this in any way, the search button will light up green to remind you that you need to push it. But if you do push that, you will see that this data will get filtered down even further to 2 epitopes. And by switching to the assay tab, we should expect to see exclusively tetramer assays, which is the case. And in this particular scenario, there are a couple of 0201 restricted epitopes that were tested positively in tetramer assays in humans. So just as I mentioned, an example where you can continuously modify and refine your queries by playing with those parameters. Similarly, if you decided, hey, I didn't like that, I just delimited stuff too far, or I'd like to swap that out for a different type of assay, it's as simple as Xing out that particular parameter and then either adding a new one and hitting search, or Xing it out and hitting search to remove that filter and go back to where you were. Okay, so before I move on to the B-cell one, any questions there that I can answer? Okay, no. So the B-cell one deals with monoclonal antibodies. And the question here is looking for influenza A virus epitopes that have been recognized by specifically neutralizing human monoclonal antibodies. So again, chance to kind of take advantage of multiple fields. There we go. Okay, so this time we're going to use the B-cell assay details, specialized search. And then along the left-hand side, we can specify influenza A virus. This is a big one when it comes to the kind of topic we were just discussing about hierarchy of organisms. Influenza is probably the most populous of the branches of the organism tree. You all know that the strains are just immensely populous, right? So this is the most broad you could do if you're interested, in fact, in influenza A. This is going to give you every possible strain, every subtype, and every potentially even isolate from which we've curated data. So host again, human, same thing there. And then in this case, this is kind of in contrast to what you see in the assay fields of the T-cell data. Under the assay fields of or assay cluster of the B cells, of course, the effector cell phenotypes, et cetera, those don't apply, but instead you'll see things like assayed antibody. So this is going to be information about isotypes, purification status of the antibodies, et cetera. But firstly, what we're doing here is we're using what's called the qualitative measurement field, which is a drop-down list, to specify that I want positives only. This is no different than the kind of default positives that was on the homepage search. And incidentally, you see that we have sort of a gradient of positivity, positive low, positive intermediate, positive high. This is strictly based on how the authors describe the data. So in some cases, they will actually compare their data and say, this had a relatively higher response than this epitope gave a higher response than this one in an ELISA or something, and we will capture that as such. But where this is probably most applicable would be in more quantitative assays where you have, for instance, a binding constant, and there are clear-cut definitions between high, low, and intermediate. But in any case, there is sort of a tree-like relationship here, because by selecting positive, you're not going to exclude data that was described as positive but weak, or positive but intermediate. You'll get it all. Okay, so moving along, what we then want to do is define the rest of the parameters, which were the specific assay type. So by now, I think you're pretty familiar with how the assay finder works. but simply typing in neutralization there would work, but by opening the assay finder, you could also do a free text search for the word neutralization. And similarly to the way the organism tree works, by searching, you can actually get results and highlight those in the tree, and you see where neutralization comes up. It's nice to kind of just explore this tree by opening up the plus signs to get some familiarity with the names of the assays. Of course, to some degree, they're standardized, but we have our own sort of nomenclature as well, so getting familiarized with those will help you, I think. But in any case, here we'll choose neutralization and hit apply. As I mentioned, you could have just typed that into the free text field, but the end result is the same, that you've restricted all the data to those that have specifically been shown to be neutralizing antibodies. And so lastly, I mentioned there's a purification status, and since the query originally intends to look for monoclonal antibodies, you can use this drop-down list to specify monoclonal. So that's, again, a multi-select checkbox. And then by running that search, you see you get 55 epitopes, but here I'm on the assay tab, and as you might expect, there's quite a bit of neutralization data out there, and especially now, as Bjorn mentioned, these are probably pretty recent publications in the last 5 years or so. And here you can see, as I mentioned before, you're not necessarily going to see strictly one type of positivity. There may be different levels. And the other nice thing is, of course, if you wanted to see the details, you could click the assay ID and see the individual field level. But it's summarized nicely even down to the strain of the flu that was used in the neutralization assay. So, this would be like an in vitro neutralization where the monoclonal antibody was shown to neutralize these particular viruses, subtypes and viruses. So it's a nice snapshot of the assay details in that case, in that sense. Yes? Go, Mike. Yeah. Okay, great. So Carrie was mentioning that in this column called antigen epitope relationship appears the phrase taxonomic sibling. And so what this column or what this field captures is essentially the relationship between the antigen in the assay and the source of the epitope. So for example, if the article or the manuscript states that we took this particular H1N1 Puerto Rico epitope and found that when we generated monoclonal antibody response in a mouse by immunizing it with it, you could take those antibodies and also neutralize an H3N2 virus, then the source of the epitope being H1N1, but the assayed organism, in this case being H3N2, are taxonomically related, but obviously they're not one-to-one. So we would say that they are siblings in terms of their taxonomic relationship. You could also have a scenario where there's a generic, the epitope is captured as a generic high-level influenza A virus without a strain. But in the assay, there is a very specific organism such as this that's been used and shown to be neutralized. In that case, we would say that the antigen was a taxonomic child of the source of the epitope. So it's the nomenclature that we use to show the relationship between the source of the epitope and what was actually assayed, because those won't always necessarily be the same. I don't know if you wanted to add anything to that.
So that's a nice way to get at cross-reactivity. So if you're interested in cross-reactivity, that's a way to look at it, using that relationship.
Yeah, in fact, everything here, well, there's one instance of child, but you can see in the epitope column that the reported epitope comes from these strains, whereas the reactivity is against these. So where do we draw the sibling relationship? Is it at the family level? So there are cases where they're both from the same subtype, H1N1, but they're different strains. We would consider those siblings.